In this video, we are going to talk about the phylogeny and what are the evidences that are used to depict or construct the phylogeny of any species or a group of species. So, what is phylogeny? Phylogeny is the evolutionary history and evolutionary relationship of a species or a group of species. So, phylogeny is basically illustrated or depicted in the form of branching tree-like diagrams that is known as a phylogenetic tree. So phylogenetic trees show how organisms or group of organisms are related to each other. So here in this figure you can see this is the phylogenetic tree depicting the evolutionary relationship between different groups of vertebrates, different classes of vertebrates, and at the base of this phylogenetic tree you can see the taxonomic characteristics that are used to construct the phylogeny. And next, what are the evidences that are used to determine the phylogeny? So first of all, fossils. Fossils have been traditionally used for a very long period of time to depict the phylogeny of different groups of organisms. So what are fossils? Fossils are basically the remains or traces of organisms that existed in the remote past. So fossils are basically present in the sedimentary rocks and in the form of layers known as strata. So the age of the rock or the strata is determined by using different radioactive isotopes, and then we can predict the time period in the geologic era when the organisms or a specific group of organisms existed in the evolutionary history or the history of life on Earth. So the arrangement of fossils in the strata gives us an idea of what organisms existed in specific geologic time. Next is homology. Homology is basically the similarity in different physical features or morphology. It is known as homology, and it is useful in determining the evolutionary relationship among different groups of organisms. So it can be divided into different types. So first of all is morphological homology. Morphological homology is basically the similarities that exist in the structures that have the basic form, similarity in the basic form, and it can be used to depict the common ancestry of a group of organisms. For example, here in this figure you can see the structure arrangement of bones in the four limbs of human, dog, bird, and whale. These are all the vertebrates, and the arrangement of bones is similar, so it can be used to depict that these structures have originated or diverged from a common ancestor. So mammals are basically classified as monotremes, placentals, and marsupials based upon how they produce offspring. As you all know that monotremes are egg-laying mammals that lay the egg, and spontaneously after laying the egg it hatches. And next, placentals, the mammals that have specific or proper placenta formation are placed in placental mammals. And marsupial mammals are the mammals that have a marsupium or a pouch-like structure in which the newborn offspring are placed. Next is molecular homology. Molecular homology, as you all know, that all of the living organisms possess biological molecules, macromolecules, DNA, RNA, and proteins, and the related groups of organisms have similar structure of genes or RNA or similar type of proteins with few modifications. So the sequence of nucleotides in DNA, RNA, and the sequence of amino acids in the proteins can be used to illustrate or depict the ancestry of a group of organisms. Next, developmental or ontological homology is that during the developmental period or development of embryo, different various stages of development are similar in different species that are related to each other. For example, the vertebrate embryos, including humans, they possess gill slits and tails during their embryonic development. So the gill slits and tails disappear in terrestrial forms, but in aquatic forms such as fishes, these do not disappear. So ontogeny is the recapitulation of phylogeny. Next is molecular biology. Molecular biology, as you all know, that DNA and ribosomal RNA are basically used for studying the evolutionary relationships of organisms. So these are the macromolecules that are possessed by every group of organism, from the simplest to the most complex, from bacteria to humans. So ribosomal RNAs are basically used to study the phylogenetic history of any group of organisms. So DNA sequencing is basically utilized to determine the evolutionary relationship of organisms by aligning the comparable sequences. Just the comparable sequences are basically aligned, and the sequences of the nucleotides in that particular gene or protein that is compared with each other.